Hi, my name is Fairlie, and today I'm going to discuss my project on analyzing the ENCODE for long-read RNA-seq dataset, which has revealed distinct classes of isoform diversity. To get right into it, alternative isoforms are different RNA molecules that derive from the same gene encoded for in DNA. And this allows both for diversification of the proteome and transcriptome, but also serves as a regulator of gene expression based on differential isoform susceptibility to regulatory processes. And transcript isoforms can be specific to different biological contexts, such as cell type specific, tissue specific, or developmental stage specific. And part of being able to understand any biological system, I believe, includes being able to annotate and determine which transcripts are expressed in that system. So the canonical view of alternative splicing is that of alternative isoforms is that they derive from alternative splicing, which is a process by which different exons encoded for in the DNA are either included or excluded from the RNA, and therefore, in the case of protein coding genes, cause for inclusion or exclusion of different protein domains, which might in turn affect the function of the protein. But it's a little bit more complicated than that. Genes can different isoforms can also result from variation at the 5' and 3' ends from alternative promoter or poly A site usage. In the cases of protein coding genes, and in some cases where there's alternative 5 or 3' end usage, we do see differences in the protein coding content of a transcript, such as in this top case here. But the 5' and 3' UTRs are also full of elements that tend to have some sort of post-transcriptional regulatory impact, such as in the case of 3' UTRs, which serve as a particularly rich binding site for microRNAs or RNA binding proteins, which are post-transcriptional regulators. To study alternative isoforms or full-length isoforms, we like to use long-read RNA sequencing. And this is because short-read RNA sequencing figuratively and literally falls short of being able to profile these full-length isoforms. Consider the case where you have these two isoforms that come from the same gene, one which contains the red and green exons and one which does not contain either. Short-read RNA sequencing can tell us which exons are right next to one another in the transcript, but cannot tell us that the red exon is always seen in the same transcript as the green exon. By contrast, long-read RNA sequencing sequences through the entire molecule, giving us not only the exon connectivity of the transcript, but also the putative 5' and 3' ends of the transcript. And this is an important resource because, as you see, many transcriptome annotations are actually incomplete. And this is partially due to their reliance on length-limited assays, such as short-read RNA sequencing or PCR. But it is also derived from the fact that the focus of these annotations is typically on the exon content of the transcripts and not as much on the 5' and 3' ends. Therefore, we think that long-read RNA sequencing should be harnessed to catalog novel isoforms and understand the transcriptional diversity in these model organisms as a whole. My lab is a part of the ENCODE consortium. ENCODE, or the Encyclopedia of DNA Elements, aims to characterize and catalog all of the functional elements in the human genome using a variety of high-throughput, typically sequencing-based assays. They can they perform these assays to profile things such as chromatin conformation, chromatin accessibility, transcription factor and histone ChIP-seq binding on the genome, but also the functional output of the human genome in terms of transcription. My lab in particular produces all of the long-read RNA sequencing data for ENCODE. So the data that I'll be working with today to show you is the PacBio human long-read RNA sequencing data on the ENCODE portal. Altogether, this comprises of 51 libraries for 10 tissues and 87 libraries for 40 unique cell lines and in vitro differentiated cells. Altogether, this is comprised of around 200 million full-length reads, and we believe that this is one of the largest and most comprehensive collections of human long-read RNA sequencing data that there is. So first, a brief overview of how we process this data. starting from full-length non-chimeric PacBio reads. First, we map our reads, and then we correct common long-read RNA sequencing artifacts, such as microindels. Then we annotate each read to a transcript that it came from, and importantly, identify novel transcripts. Then we use a new tool developed in the lab called LAPA to call 5' and 3' ends from our long reads and filter these 5' and 3' ends based on their reproducibility and external validation of other transcriptional assays such as CAGE or Rampage or PAS-seq. Then we use another new tool developed in the lab called Cerberus to kind of create a transcriptome that's the result of integrating a lot of different transcriptome sources, such as multiple annotation versions of the annotation, our long read data, as well as these external sources supporting the existence of 5' and 3' ends. So first off is to check what we see that is annotated already in our annotation. So if we compare our what we detect to the GenCode 40 annotation, we detect around 65% of polyadenylated genes, which is a category that's important to limit ourselves to as we do perform an oligo-DT selection step in our library prep. But when we narrow this frame to only look at protein coding genes, we actually detect around 94% of those. So a pretty hefty increase. On the transcript level, we detect less, which makes sense because it's easier to detect a gene than it is to detect a transcript. But we still detect around 32% of polyadenylated transcripts and 38% of protein coding transcripts. But what about what we detect that is novel? Because again, if we're going to try to supplement our understanding of the human transcriptome, we need to be able to find what's there that's not annotated already. And to this end, we have this method of identifying, but also classifying novel transcripts with respect to the reference genome. So novel transcripts can be one of 3 different novelty categories. The first one is ISM, which represents a subset of a novel or of a known transcript, such as these 2 transcripts seen here. We can also have novel in-catalog transcripts, which are characterized by the presence of a novel splice junction, and novel not in-catalog transcripts, which are characterized by the presence of a novel splice site. All of our novel transcripts are filtered for reproducibility, but the ISMs are subject to a bit more of scrutiny because they are oftentimes just the product of degradation or incomplete sequencing through a molecule. In the case of ISMs, not only are they subject to reproducibility filtering, but we also filter them on the basis of internal priming. And we require that the 5' and 3' ends of these transcripts have external validation from CAGE or PAS-seq. And altogether, this means that we find about 86,000 novel transcripts for a total of around 220,000 complete detected filtered transcripts, including both our known and novel. And this is what the breakdown of them by their novelty types look like. So the majority of what we detect is known transcripts, which is, you know, a good thing, a good sanity check. So now what we can do is we can ask ourselves, how do our number of isoforms differ for each gene when we compare to the number of isoforms that are annotated already in GenCode? And that's what you're looking at here. So on the y-axis is the number of isoforms in GenCode, and on the x-axis is the number of isoforms that we identify in our long-read RNA sequencing data. So anything that falls above this x equal y line is a gene that has more isoforms annotated in GenCode than we see in our data. Whereas below the x equals y line is vice versa. We see more isoforms in our data than are annotated in GenCode. And I've just pulled out some interesting examples here, first of which is MapK10, which has the most isoforms annotated out of every gene in GenCode at 192. But also 2 genes that have a pretty marked increase in terms of their transcriptional complexity between GenCode and what we observe. So elastin goes from having 35 annotated transcripts to 286, and Desmond goes from 5 to 55. So next, before I show you an interesting result, I'm going to go over a new method of naming transcripts that we're using to try and distill the information content about how the transcript is structured for each transcript. So bear with me for a moment. Okay, so suppose we have this isoform here. we are just going to say that each isoform from a gene has a transcript start site, a TSS, it has an intron chain, and it has a transcript end site, a TES. And so if this is the principal isoform for the gene, which is annotated typically in your reference transcriptome, we will just go ahead and say that these are the first intron chain, the first TSS, and the first TES for this gene. And so we can give this isoform a name which uses the numerical identifier for each of these features now. So we're going to just call this gene, very creatively, gene 1 for the first transcription start site, 1 for the first intron chain, and 1 for the first transcription end site. Now, as we add isoforms, we can increase these numbers as we see new unique features to this gene. So if we add this transcript, which includes a new intron chain and a new TES, we can now call this gene 122. And finally, if we add this last transcript here, which introduces a new TSS, we can call it gene 221. And this naming strategy allows us to easily compare the content or structure of each transcript within a gene just based on their names. So just from this, you can see that these two isoforms here share a TSS because they have the same number in the first number right there. And similarly, for these bottom two genes, we can tell that they share an intron chain based on the second number. And so then we can go ahead and do things like this with this strategy as well, which essentially defines the group of transcripts that from this gene that all use the same TSS, or the group of transcripts from this gene that all use the same intron chain. So using this strategy, which again is just for display purposes currently, I just want to show what the relative expression of each isoform in MEF2C looks like across our data set. So as promised, each of these transcripts is named in the prior convention that I just mentioned, but these are also the isoforms that are expressed in MEF2C in these data sets. And the color indicates the relative expression of that isoform within the gene, where it goes from 0 to 100, where 100 indicates that the entire gene's expression is from that one isoform. And what you'll notice is that in these 3 cell lines, all of the gene's expression comes from this one isoform of MEF2C, which is truncated. And interestingly, MEF2C, which is the transcription factor, has this transactivation domain in the 3' end of the transcript typically, which this isoform notably lacks. And for those that may not know, the transactivation domain of a transcription factor is responsible for binding and recruiting external factors that come and help the transcription factor enact its regulatory regime within the cell. So this is likely an isoform that is either non-functional or less functional than the other isoforms expressed here. And it is the only isoform that is expressed in these 3 cell lines. So there's something interesting going on here. Okay, so this is a nice little example, but we have a huge data set and we want to be able to say something globally about the transcriptional diversity in our data set. So another kind of teaching example here. We want to be able to essentially, instead of making an isoform triplet to indicate the name or identity of each isoform within a gene, we also want to be able to compute a gene triplet, which indicates kind of the diversity within the gene of these 3 different features that each isoform can have. And so here we can identify each gene using an isoform or a gene triplet, which tells us how many TSSs are present out of all of the isoforms in the gene, how many intron chains are present, and how many TESs are present. And so this gene here, this top one, which has 1 TSS, 2 different intron chains, and 1 TES, can be described using the gene triplet 121. And down here on the bottom, where this gene has 4 unique TSSs, 2 unique intron chains, and 1 unique TES, we can describe this using the triplet 421. And using this strategy, we can actually now go and display how these genes how these genes triplets differ from one another on what's called a simplex, which is this triangle here. And I'll just go over where these genes will lay on this plot. And you'll have to trust me on that for now. But essentially, what's going to happen is genes that fall within this bottom left sector will be genes where the majority of the genes' isoform diversity comes from variation at the TSS. Genes that fall within this top pink sector here will be genes whose isoform diversity mainly comes from variation in internal splicing. And genes that fall within this bottom right sector here will be genes whose variation predominantly comes from, diversity predominantly comes from variation at the TES. Genes that fall within the center of this simplex will have one of two fates. The first of which are genes that only have one isoform, which we lovingly call simple genes. But for anything else that falls within the sector with more than one isoform, we will call it a mixed gene. And so here's just a little set of toy genes that might fall into each of these different sectors here. So our pink gene has two different intron chains. Our orange gene has two different TESs. Our blue one has four different TSSs. And then our mixed and simple genes accordingly only have one or two isoforms. So what does this actually look like in practice? Well, here we're comparing the density of these genes based on their transcriptional diversity, based on those triplets, between GenCode on the left and between the data that we actually see in our long-read RNA sequencing on the right. And so what you can see here is that we actually see kind of like an outflow of our genes into these corners of the simplex compared to GenCode, especially at the 5' and 3' end. We see an enrichment here compared to GenCode, which is not surprising for reasons that I already mentioned, which is basically that we actually pay attention to the 5' and 3' ends of our transcripts now, as opposed to GenCode that only ever annotates a 5' and 3' end whenever it comes along with a new intron chain. So we can also plot these simplex plots for the sets of isoforms that are expressed in each of our different samples. And so that's what you're seeing here. On the left is elastin, and on the right is MEF2C. And each of these colored dots here indicates the triplet underlying the expressed isoforms in each of these different samples. And so in the case of elastin, it tends to, in most of its expressed samples, exhibit a high degree of internal splicing. Contrast this with MEF2C, where most of the samples that it's expressed in exhibit a high degree of alternative TES usage, which is consistent with the example that I showed earlier, which resulted in that truncated MEF2C isoform. So we can also limit the isoforms that are used to calculate these within sample numbers, within sample gene triplets. So on the left here is the normal plot using all of the expressed isoforms for col3A1 in each sample. And on the right are the same triplets, but this time calculated only using the major isoforms expressed in each sample for col3A1. And we're defining major isoforms here to mean the set of isoforms that cumulatively are responsible for over 90% of the gene's expression. And so what you're really seeing here is that col3A1 has a lot of minor isoforms that aren't very highly expressed at all compared to the major ones that typically display a large degree of internal splicing. However, there are only a few isoforms that are actually responsible for this gene's expression. And in the cases of these isoforms, they typically vary either at not at all or just at the 3' end of the transcript. So this is an interesting shift that we're seeing based on the relative expression of these transcripts. What we can also do is compare the localization of these genes in the simplex based on what species that we're sampling. And so for this case, I'm limiting on the left in human to just the samples that kind of match what we have matching data for in the mouse. So here we're seeing TCF3 and on the left is human, on the right is mouse. And you can see that in the case of human, for the most part, our samples lie within this internal sector, our mixed or simple sector, but all of them exhibit a really, really high degree of internal splicing in mouse. So there's something interesting going on between human and mouse here. But of course, we can also find instances where things look the same between human and mouse, such as in the case of Col1A1, where most of our samples have diversity limited to only the internal sector or the TES high sector in both human and mouse. And as a broad overview of this comparison between the species of the genes that they share in common, around 42% of those genes sectors are conserved between human and mouse. So in the future, what we're interested in doing is a couple of things. Firstly, we want to come up with a way to represent genes in the simplex based on the relative expression values of their constituent isoforms. So this would be kind of an alternative to using just the major isoform set that we used in the Col3A1 example. We are also interested in characterizing the functional potential of our alternative isoforms, mainly in terms of the protein coding potential, as well as in microRNA binding site inclusion. And finally, we want to do more of the sort of like analysis that's like, how do these genes shift from one sector of the simplex to another between different biological contexts? So I showed you a brief example between species, but we'd also like to do this maybe between samples, but also between all and major isoforms. And with that, I'd like to thank a lot of people. I do consortium science, and it truly is a team effort. So if your name is on here, thank you. But if it's not and you're listening to this, I also want to thank you for your time. And I hope that you feel like you learned something.